Well, hi everybody. Thank you for showing up so early today. We're probably going to have a relatively brief session given that we have only so much time in this room. But I would like to go ahead and get started with our gene expression study. In this case, we are going to focus much of today on a web resource called Gene Expression Omnibus. The Gene Expression Omnibus, or GEO, is a really helpful repository of data sets that have been published in the space of gene expression. A lot of people still think of it as that place where you can go find microarray data sets, but the reality is an awful lot of expression data, up to and including things like RNA-seq and even methylome data, can be found at Gene Expression Omnibus as well. So even though it may have started as what was heavily a microarray resource, today it's very much gene expression on a much broader scale than that. Okay, so we're going to start with a data set. Frequently, we will work with data sets that are represented in repositories by way of an accession number. So in this case, we see GSE 43837 is where we're going to start. I'm just going to go ahead and click that link. Hopefully, if the web is behaving itself today, we will get a helpful display to work with. So we see that we have a title, a Brca1 deficient-like signature is enriched in breast cancer brain metastases. Now, I'm accustomed to working in cancer research. I realize not a lot of you would be, so let's talk a little bit about this. First off, has anyone heard of the Brca1 gene? Good, yes. Brca1, yeah. So Brca1 is one of the genes that is most strongly associated with breast cancer. It may surprise you then to learn that most women who develop breast cancer do not have a faulty Brca1 gene allele. So it is associated strongly with breast cancer. People who do have double hits on this gene are quite likely to develop breast cancer, but it is not the causative agent in most cases. Okay, so Brca1 is important here. Next, we've got brain metastases. Brain metastases. So how do people get breast cancer in the brain? Okay, so this is one of the most important things to understand about cancers. Generally speaking, if cancers can be found in their initial locations, I guess you'd call it the primary lesion. We use the same kind of language when we talk about tuberculosis. If you can spot a cancer while it's still in its primary lesion, you have a pretty good chance of being able to sequester it from spreading throughout the rest of the body. Because one of the things that cancer is so evil about is that it diminishes a cell's responsiveness to the stimuli that usually keep that cell in line. You see, our cells don't just split and split and split endlessly. We're not bacteria, right? Our bodies have developed these mechanisms such that the cells divide until they fulfill their developmental niche, and then the pressure of cells around them is one of these triggers that says, okay, stop dividing now, stop growing. So a tumor cell, on the other hand, loses this ability to communicate with the cells around it and decides, oh, I'm just going to grow crazy-like. So it does things like recruit new blood vessels so that it gets more oxygen. It divides more frequently. It loses a lot of the proofreading machinery, which means it can develop even more mutations. And sooner or later, it develops the ability to abstract itself from its initial environment, maybe go through the blood system a little bit, and end up in a completely new part of the body. If it ends up in your brain, this metastasizing cell can set up shop as a new secondary tumor in a new part of the body. And of course, if you've got the tumor growing in your brain, it's a little harder to get to than if it's in a conveniently located external location. So this is one of the reasons why we talk so much about screening for breast cancer. Because if we detect a tumor in the breast, we can get at it pretty easily, not necessarily for the person who's experiencing it, but we can get it. And that prevents it from launching off these little colonies off in other parts of the body. Okay, so when we talk about brain metastases, we're talking about cells that have left the breast, gone through the circulation, maybe they've gone through the lymph nodes, and they've shown up in the brain where they've set up shop to create secondary tumors. Great, okay. Now I had a series of questions I wanted to ask about these. You see we've got these 5 right here, so we'll just work our way through that. On what date was GSE 43837 submitted? All right. Okay, it went public right there. Public on April 24th, 2014. Great. When was it submitted, though? Where'd you find it? Ah, there it is. Submission date, January 28th, 2013. So that's a rather long time between them, isn't it? Frequently, we will find at repositories that people submit data, and then when the manuscript is finally accepted, they will say, okay, now it can go public. Right? So the repositories contain many entries that have not been made public, in general because people are waiting for the acceptance of the paper. There are some people who are not required by their funding agencies to make those data public, and they just use it kind of like a big file server in the sky. But the purpose of repositories, generally speaking, is to make your data available to others. I'm not going to be spending a lot of time talking about the FAIR principles, but you can look up terms like open science and FAIR, and you will see some of the principles behind why reproducibility is assisted by people making their data public in this way. Great. Okay, so how many samples are involved? And along with that, what are the cohorts? All right, who wants to start on this one? I'm kind of a sucker for free text, so I'm going to look at this, this big summary section. Look under experimental design. Gene expression of 19 HER2 plus breast cancer brain metastases was compared with HER2 positive non-metastatic primary tumors. Gene set enrichment analysis was used to identify a signature, yada, yada, yada. Okay, so that to me is suggestive that we have a total N, if we count all of the different samples, of 38. Is my math working? I'm a little sleepy. Anyone else sleepy? This is a good nap time, isn't it? It's a shame we don't get our afternoon naps, but okay. So we have breast cancer brain metastases, and we have non-metastatic primary tumors. I'm going to suggest that we've already encountered something that should cause a slight eyebrow raise. Right there. An eyebrow raise, because they are comparing breast cells to breast cells. That's cool. But in one case, those cells are in the breast, and in the other case, they're in the brain. You might imagine that a cell that is moved to a new location is going to behave differently, considering the signals that its neighbors around it are giving you. So we may think that these cells are changing their behavior strictly because they are in a primary tumor or in a secondary tumor, but they're also getting different messages from the environment around them. Okay, so we have two cohorts. We have 19 in each. All good so far. Let's return. What kinds of microarrays are used? All right. Well, it says by array up at experiment type. That's not so useful. What if we drop down to the bottom? Design. Oh, here the study design says that 19 and 19 thing. Right all over again. That's good. All right. Anyone see anything useful? Ah, someone's got it. Okay, good. We have platforms mentioned here. This is brilliant. Oh, and the total number of samples is given in numeric form down here. 38 samples. Great. So we see that this was collected on a GPL 1352. Many repositories make use of controlled vocabularies. Controlled vocabularies show up in a lot of places, so I'm mentioning it here. We see that they have a particular kind of device, this U133 array that's sold by Affymetrix. Affymetrix sold a lot of gene chips. Gene chips are these microarray designs that that were so very popular. So we see that the U133 platform was used. If we click through on this link, we'll get more information about what that accession means from the controlled vocabulary. So I click it, and I see that I get a whole bunch of information here. So we had some information, I think we were asked, yes, how many different probes were measured on each array? I'm going to click back. We're not on the Brca1 page now. We're on this particular platform from it. So we see that it is designed for use in Homo sapiens. Well, that's good news, right? These are humans who had breast cancer. It could be a breast cancer study in mice. You know we do that, right? Has anyone heard of a xenograft? Xeno, xenograft? It's not pronounced with a click, though that would be pretty cool, wouldn't it? Xenograft. Sorry. my clicking is terrible, I'm so sorry. Okay, xenograft. A xenograft means that we have moved a tissue from one species into another species. If you're watching an Aliens movie, they may refer to aliens as xenomorphs. Xeno does not mean alien in that way, but that's fine. So if you have a cancer in a human being, it's obviously not a place where you can do any kind of experimentation, right? That the ethics required for doing human cancer studies it can be quite onerous. So if you have a particular breast cancer that you think is biochemically interesting, you may try isolating some tumor cells from the human and inject those into a mouse. The mouse then develops a tumor, and, well, ethics procedures are a little simpler where mice are involved. So here we see that instead of doing this xenograft trick, working in mice with human cancers, now I want you to think about the fact that if you did this in a xenograft, you would have both human and mouse genes in operation at the same time. The environment of the tumor would be mouse. The tumor itself would be human. Wild, huh? Working in xenografts has a lot of complexity. That's fine. But here we're looking at human gene expression. This array was made by Affy. We can go to the manufacturer's protocol and see it on their website. Here we get a little blob of text that tells us what they are. But down here, we have this bit of information about the human genome U133 plus 2.0 array. So we see that we have a total of 47,000 transcripts with 61,000 probe sets. Well, that's a little puzzling, isn't it? Why are those numbers different? 47,000 transcripts. Is that the complete human transcriptome as we think of it today? Got a head shaking no, but she's not sure. Somebody want to back her? Come on. Have your classmates back on this one. Oh, all right. Fine. Okay. We would say 47,000 transcripts is a little low. Since this since this gene array was created, studies like ENCODE, ENCODE, have done a lot to increase our experience of all the different transcription events that take place in humans. So 47,000 is actually kind of smallish. Now, why is the probe number different than the transcript number? There are several reasons why this might be the case. In the very old days, a lot of gene chips would use what are called mismatch probes. So for every probe that you had that was a perfect match for a transcript, you'd have another probe that was off by one. It would have a one-letter change in that probe, the goal being to see how much hybridization took place when there were slight sequence variations. It was one way to evaluate the background noise. But here, it's quite likely that they have probe sets that are specific to a given gene and others that are specific to a particular transcript. Do you see how you might want to have one probe that would light off for every transcript known for a gene and another one that was specific to which transcript was being produced by it? That's kind of cool. So there's that, and we probably have some degree of redundancy anyway. They may know that some of these gene probes that they're using are a little iffy, and having a backup one is useful to get a nice, reliable signal for a particular gene. Okay, great. So we have all this information now about that. This information's all been gleaned out of a text file from 2004. 2004. So now we're accustomed to thinking that the technology couldn't possibly be worth anything unless it's today's technology. But here, for an analysis published in 2014, the microarray was first released in 2004. So that is an interesting factoid. All right, now I'm going to come back here. we are going to do a reanalysis of these data. So the software gives us the ability, I'm going to back up one page here. That should put me right back on this page. I'm going to drop back in size a little bit. Okay, so this is what the whole page looks like for the Brca1 thing. Down at the bottom, did you all see that big blue button, Analyze with Go2R? Okay, so one of the coolest things about today's repositories is that many of them have what we call reprocessing capability, which is to say that they're not just big file servers in the sky. They're file servers that have tool sets built into them that allow you to reanalyze the data sets. So when this study was conducted, many of the tools in Go2R may not have existed. So we're going to use the tools this web server, this repository considers appropriate today to analyze data that were collected in the past. One of the consequences of this is that we may come to a different conclusion about what these data mean than what the authors did when they published it. We're going to get different results. I'm almost sure that the p-values and full changes that they reported in their paper are going to look different than the ones we're going to get out of this pipeline. Weird but true. Okay. Which means that what you can extract from the data you generate today as an honors student may change by the time you get a PhD, if that's where you're headed. Weird. Okay, so let us now click Analyze with Go2R. Click, click, click. All right. Great. Look at all this good stuff. Did anyone's link not go through? Well, that's still so lovely. I'm glad you're following along. All right, so we said that there were 38 of them here. And I can scroll up and down a little bit. Zoop, zoop. It all looks good to me. These are all supposed to be HER2 positive. Does anyone see an exception? This is this column right here. HER2 positive. HER2 positive. That looks good to me. I see a bunch of HER2 positives. Good. Did anyone see any information to help you separate these into the two cohorts they mentioned? You should see a slight change right about there. You see that? Brain metastasis, primary breast tumor. Remember, these are both enriched for breast tumor cells, but some of these have been found in the brain, which means they migrated out. In my reading of South African history, I think it's really interesting that every town you go to in South Africa has a Voortrekker road somewhere. And one has to imagine that not every Boer was equally likely to go on the trek, right? There are some that are curmudgeons who are just like, nobody can tell me what to do. And you can easily imagine that one heading out of town, right? That's a Voortrekker. but there have to be some number of Boers who were like, yeah, it's okay, I like it here. So in the same way, you can imagine that these breast cancer cells, some of them are particularly adventurous spirits. They're the type that are kind of melting away the integument that holds that cell in place in order to break free and go move through the circulatory system. And others don't, right? So some cells are particularly prone to this because they've racked up enough mutations that they behave in ways that normal cells won't. All right, great. So we now need to do a define groups process. If we click back over here, we see that we must define two groups, one called metastatic, one called primary. So I have to remember how to do this. I come back in here. I'm going to move to the top of this little table. I'm going to click this one. I'm going to click that one. Oh, no, see, that didn't work. See, when I clicked the first one, it was fine. But when I clicked the second one, it clicked away. So I'm going to try something else. I'm going to, let me go ahead and blow out define groups here. I'm going to give this one a name. I said metastatic, right? Metastatic. All right, I'm going to give another group name. These are primary. Okay. Grandola. Now, let me see if I remember how to do this. I'm going to click on this first one. I'm going to roll down the screen a little bit. And here's this last one. Don't click on it just yet. Hold down Shift and click on it. Oh. So it is possible to hold down Control and click each one progressively, but this is a little faster. Now, while you got those clicked, I'm going to come up here and specify these are metastatic. You see that? Now it has a group label attached to all of them. Similarly, I can come down here, roll to the bottom, hold down Shift, and click the last one. Boop. Now I can click on primary. Okay, so at this point, I should have all 38 files labeled either metastatic or primary. Did that work for most folks? Okay, holding down Shift is kind of it's a trick for people who've used a whole lot of different Windows tools, but hopefully it's a useful one for you here. All right, so we have now defined two groups. We've assigned all the brain metastatic breast cancer rows to the metastatic group. We've assigned all the non-metastatic breast cancers to the primary group. And now we want to examine the value distribution plot. Okay, I was very sure how to do that a couple years ago, so it must be pretty straightforward. Ah, look at this. All I need to do is click value distribution and view. It's passing up to Europe, it's passing across the Atlantic, it's coming back. It's waiting. It's running. You know, if we all click view at the same time, that server's just like, dang it, it's another class. It's usually considered a nice gesture to email the repository the day before you run a class. I did not. So I'm the bad boy here. All right. So now we have a value distribution plot. What on earth are we looking at? Anyone? What type of plot would you call that? A box plot. Thank you. Good. You've seen these before. The box usually means the middle quartile, the interquartile. So you can say that the 25th percentile is at the bottom of the box, and at the top of the box we've got the 75th percentile, and then the stripe across it represents the median, typically. Okay? Well, we've got some extensions on here, right? So what we're looking at is the distribution of intensity values for each microarray. But there are 60-some thousand intensities here. So this is a distribution that's got a whole lot of points below it. All right. I think it's important that we ask a few basic questions about this plot. How would it affect things if we had a high degree of variability within a cohort? Let's look at just the blue ones, the metastatics for the moment. Okay, so we're only paying attention to the left half of this plot. How would we feel if we saw that some of these boxes were tall and some of them were very narrow, and others had median values way up high and others had median values way low? So what would you call an abnormally high-looking one? Maybe this guy right here? It's got a median that's much higher than those around it. But maybe if we sorted all of them by median, it wouldn't stand out quite so much. Kind of hard to know. Okay, so what hopefully everyone remembers from the biostatistics module is that the rate... Why does everyone laugh whenever I talk about the biostatistics module? I'll just repeat what everyone, of course, learned from the biostatistics module. That if you're looking for a difference, the amount of variability you've got matters. Because if that difference is very small, you need a very, very tight reproducibility, a very low variability in order to find it. If the difference is huge, well, you can get away with a lot more variability. So generally speaking, we would like to think that the variation we see is not a huge factor. What we see is that from a normalization perspective, there's probably a bit that could be done to make these data look more like each other across the board. Hopefully the software is going to have a bit of a normalization model internally that will take care of that. All right. I'm going to run myself right out of time. I like talking too much. Let's come back to this guy. All right. Do you see any bias towards a higher signal for one of the cohorts? All right, so think about batch effects. Did they mention the word batch effects at some point? Oh, that's good. That's comforting. Good, good. Okay, so what would happen if our microarray had good days and bad days? I know I'm talking about the equipment as though it has a personality. Trust me, it does. All right, so it may be that our microarray equipment is feeling really punchy some days, and other days it's like, whatever, man. Right? So some days you might have a really hot, if you were to run all of your metastatics this week, when the instrument is like, you know, ready to fight fire with fire, and run all of your controls the next week when it's just barely turning out a signal, you got a big problem. Because now you have an instrument effect commingled with your cohorts. It's really, really bad. This happens a lot in biotechnology. You would be surprised how often people don't take this into account, and they end up with egg all over their face. It's terrible. So, in this case, we look at the data and we ask the question, do we see an obvious difference between the pink and the blues? They both spin around a little bit, right? They both skitter a little up and down, and on the whole, I would say those median values are jumping around a little bit more than I would really, really like. Be aware that most published data have something a statistician can object to. It's true. We would kind of like to see a little less dancing around in the variability of the raw signal here. Does that mean that this publication should never have gone forward? Probably not. That's not where we're going with this. I'm just saying that when you look at the data from a QC perspective, a quality control perspective, you'll frequently find there's something there that makes you twitch a little bit. So this eye for me, it just kind of goes twitchy. Okay, so now let's come back to this. We are going to produce a top 50 report. So I come back to this interface. I come back to the Go2R tab, Gesundheit, by the way. And I'm now going to click on the top 250 report. Everyone sees that? All right. What is the top 250 report? To perform the calculation with default settings, the top 250 genes are presented and may be viewed as profile graphs. Well, that's lovely. Great. Let's do it. I read the directions, don't you guys? You read the software manuals and stuff like that? That's what they're there for. I just say, as a software developer, if I write a manual and somebody emails me with a question that's in the manual, oh, yeah, exactly. Read the manuals. It's very, very good. Good. At least the paper that introduced the bloody thing, you know, you owe it to the developer. All right, there we go. Great. So we now have a bunch of IDs, edge, p-vals, p-values, T's, B's, log FCs, gene symbols, and gene titles. Brilliant, right? All right. We had some questions about these, I think. What type of normalization was used with the raw intensity values for each probe? Anyone see it? What is intensity normalization? Why do we care about this? I think you saw a video about that last night, didn't you? Oh, it was that was the YouTube video you missed?
No,
oh.
I watched that. That was the second one.
Someone remembers there was something about normalization in there. We were just looking at a value distribution plot, right? And we said, gosh, I sure hope some sort of normalization takes place that deals with the fact that some of these microarrays were more responsive than others, right? So some sort of normalization has been used, and if you look at the very top line on screen, it says what?
Log transformation.
There it is, log transformation, brilliant. Okay, log transformation is a very, very typical approach used by lots and lots of people. I know that it is not Gerard's favorite, which is hypergeometric arc sine, whatever it is. That's fine. It's another standard approach that lots and lots of people do. Log means that the really huge changes in magnitude will be ironed out to a very great extent. Great. Let's come back to this. All right. Why would the p-value and adjusted p-value columns be so different? So first off, what's the table sorted by? Probably the p-value?
Adjusted p-value.
By the adjusted p-value? Are those orders the same? Yes and no. I'm seeing heads shaking and people saying yes. Okay. Well, I would just point out that adjusted p-value here is rounded to 3 decimal places. So we have like 205, 205, 205, 205, for example. Okay, so the orders should be the same in this case. All right, so why are these two values different in this case? Nothing. I wrote a whole video about this, the very last one you watched last night. Multiple testing correction. Multiple testing correction is very, very much needed here. If you are working on a probe-by-probe basis, and it seems like each row here represents a different probe, it would appear that we must use multiple testing correction because we have generated a huge number of statistical tests. So when we include adjusted p-values here, it reflects the fact that we needed to adjust for multiple testing correction. That's what differentiates an adjusted p-value from a p-value. Sometimes you'll see a column that's called a Q value. If you do that, that would be a Benjamini-Hochberg FDR correction. All right, let's return to our list of questions. What type of multiple testing was applied in this set? We just inferred that these two columns are different from each other because of multiple testing. So what was used? Is it up top? I don't see it. I'm going to drop down. It's 250 proteins, right? Any stuff here? Gosh, if only there were some methods here. Where do you think it is? Tell you what, I'm going to click over to options and see if I get something useful. Options. Oh, look at that. The options tab was hiding underneath all that. What did we use in this case? Benjamini-Hochberg. Benjamini-Hochberg is like a biologist's best friend. You should just get used to seeing BHFDR. It is a really, really big friend. One of the reasons why Benjamini-Hochberg is so bloody popular among biologists is that it controls the rate of false discoveries. We talked a little bit about Bonferroni. Does everyone remember that? We talked about it with the Manhattan plots. Bonferroni is trying to protect you from making any errors, making 0 false claims. But Benjamini-Hochberg is so much gentler and kinder. It's trying to say, what is the rate at which we are accumulating false discoveries? And so in this case, using Benjamini and Hochberg's false discovery rate, we're trying to limit the rate of false discoveries here, probably to not worse than 5% or something like that. So of every 20 genes that the software says are differential, 1 is likely to be a false finding. Everyone got that, right? So you're not, once you've used Benjamini-Hochberg false discovery rate assessment for controlling multiple tests, you're no longer protected against making any errors. You have some acceptable rate of errors. So if they used a threshold of 0.05 for the BH adjusted p-values, that would mean that 1 in 20 of the resulting claims about difference would be bogus. Okay, great. So we've got our answer from the options tab. Right. Why is the ID of the probe different from that of the gene symbol? What am I talking about? ID of the probe. Okay, so the gene symbol is way over here. Do you guys know how complex the process was that ironed out what gene symbols shall be used for all 20,000 human protein coding genes? It was long, laborious, and painful. But the Human Gene Naming Consortium has this final recommendation, and everybody follows it perfectly, I'm sure. And that has been very useful for making it so when you publish a gene, publish your analysis of collagen 10A1, people have some notion what gene you're talking about. So it's actually quite valuable that this gene symbol argument has been resolved so nicely. But this is obviously not a gene symbol. What happens if I click one of these? Ah, look at that. That's cool. All right. So now we see that having clicked on the probe ID down at the left, the software sees, this one is associated with collagen 10A1. That's great. Are we guaranteed that that's the only probe that relates to that gene? Probably not. We could, in fact, sort that table on gene symbols and see if we get any collisions where multiple probes show up for the same gene. But in this case, we are looking at the values that were recorded for this probe for all of these different samples. We have a total of 38 samples. We're going to get all 38 measurements for this particular probe. That's pretty impressive, what I would call drill down, the ability to start from a summary table and work all your way back to the original data that we started with. Grand. Okay, great. ID in the probe. Do you accept the argument that many genes change expression between these two cohorts? The authors got some significant p-values out of this, right? They made the claim that they see expression differences between metastatic and primary breast tumor cells. Do you believe it? Why don't we start with the most extreme p-value we've got, right? It's all the way at the top. Remember, it's sorted from low p-value to high. So we'll just look at this one up here. All right, there we go. Do you accept that this gene, hydroxysteroid, whatever it is different between the primary and the metastatic cells? Okay, so we might ask ourselves some basic questions here. If I have to do a difference test, one of the things I want to know is the highest value of one cohort lower than the lowest value of the other one? Are these distributions overlapping? So what's the highest one we have of metastatic? The penultimate one, is that what you said? the next to last sample in metastatic has a bit of a road bump there. Boom. Now, is that lower than the lowest of the primary set? It's not. Okay, so already we have some amount of overlap in this distribution. There's at least one value from this cohort higher than the lowest of the other. So we have to have some sort of difference metric to tell us whether this is different or not. So what do we have to give us that information? We have a t-statistic up there, right? So some form of student's t-test or Welch's or maybe even a pooled variant of t-test has been used here. It produced this t-value of 5.35, and the p-value associated with it is way low, way low. But what about the adjusted p-value? After we deal with the false discovery rate assessment, does this pass muster? She's shaking her head no, but she's not sure. Back her up, or don't. Are you going to repudiate her? She sits right by you. This is so rough. All right, you can settle it after class. I'll turn away. Sorry. All right, so we have a great looking p-value in that column, but the adjusted p-value is 0.112. It's well outside 0.05. If you were to accept this as a true difference, you would be wrong 11.2% of the time. Yeah, harsh, right? Okay, so what we're learning is that this data set may not beat multiple testing correction for real differences. The software is just kind of uncomfortable about having that much variability on the right. All right. Remember that if you're going to claim a difference, you want the magnitude of your variability to be smaller than the difference you're trying to spot. The biggest differences are easiest to find, and are most robust against variation. But if you've got a very small difference you're trying to pull out, that's harder. Okay, so we're worried about this adjusted p-value now. Maybe the findings of this paper require a little bit of scrutiny. Maybe we would want to pull this data set together with others before we jumped to the same conclusions as the authors. Happens a lot. Okay, expand the information associated with the lowest p-value we just did. Are those intensity values consistent with the judgment produced by statistical testing? Well, they look kind of different. But ultimately, there's something that Gerard likes to mention quite a lot about this, which is to say that when we use models like t-test, we need to have differences, we need the residuals of the models to be normally distributed, which may mean very, very little, but I'm going to try to walk you through it. Let us imagine that the model that we're applying to these data is effectively drawing two lines, right? We have a mean value for the blue cohort. We have a mean value for, oh, the red cohort was higher, right? Sorry, so we have a red line and we have a blue line. These reflect the model that the system has inferred from the data, this being a mean from the blue, a mean from the red. So the software is evaluating this model in a way that requires that the residuals, which is to say how far each of the red points is from that red line, and each of the blue points is from the red lines, that if you look at those residuals, those differences, they should be normally distributed. I think what we're running into in this case is a problem where those residuals are not, in fact, normally distributed, which means that the t-test falls apart. T-test is not designed to work in scenarios where we have residuals that are this lopsided. We have just what the heck is exactly wrong with this third red sample? What is up with that? It's right off the charts in comparison to everything else. This is an example of non-normality in the residuals that makes me feel that we really can't stick with t-test for a data set like this. Can anyone give an example of a test that might be a little more robust for data like these that are a non-parametric test? Okay, maybe someone remembers U-test, Mann-Whitney-Wilcoxon. That's an example. That's a rank order one that we could use in a case like this. It's kind of a mouthful. The MWM is not the most articulate of names. Mann-Whitney-Wilcoxon. Okay, so we had some alternatives, but the software was designed to just apply t-test in this case. It might be that we could rescue our results if we used a test that was more suited to the variability in this study. Okay, right. You can, in fact, look at the R code. Now that you've been expertly trained in R, you are able to look at anybody's R code and immediately figure out what it's doing. The R script, oh, don't shake your head. Don't you? Yeah, there we go. That's it. Good. All right. You can see that the R code that generated those reports is right here, which means that if you were using this code to dress up the data and your advisor said, no, I want the label over here, you could just fix that line and alter the graph to your liking. So that's useful. Ordinarily, I would have you dig through the remaining questions on the document, but I think we're probably already up against our time boundary. So I hope that this has been useful. You can look in a repository, find a data set, reanalyze it if the repository offers functionality like that, and even evaluate, do you support the claims made by the authors in their publications? You, as a growing scientist, are empowered to do that. You are expected occasionally to do that. One of the uncomfortable things that you're going to realize the longer you spend in science is that not everything that passes peer review is necessarily equally trustworthy. So when you are figuring out, how am I going to spend the next year of my life? Am I going to be chasing down the results of Professor X or Professor Y? You will have the ability to reanalyze that person's data before you go about trying to acquire your own, to chase after something that's ephemeral and was a false finding. That is an uncomfortable reality, but I hope that it's one that you feel a little better prepared to take on as we move ahead. Okay, with that, I'm going to shut down the recorder, and I will look forward to seeing you. Tomorrow is Friday, right? Tomorrow morning at 9:30, right back here in the fish tank, when we will have a fun discussion of proteomic identification and quantitation, the best field in the whole wide world.